Bismillahirrahmanirrahim. Honorable Director Sir, respected teachers, dear colleagues and beloved interns. Assalamu alaikum and good morning to all. I am Dr. Maria Hussain, Assistant Professor of Physiology. Welcome you all on today's seminar on cell-to-cell signaling intercellular communication. On today's occasion, our chairperson is Professor Shahnaj, Professor and Head of the Department of Physiology. And we are lucky to have Professor Karim as our resource person today. I would like to request Sir and Madam to come on dais and conduct the rest of the session. Madam, good work.
Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to today's seminar on cell-to-cell signaling and intercellular communication. Today's speaker is our beloved Dr. Farhana, Assistant Professor of Physiology. She has been first MBBS from Select M.A. Usmani Medical College and her completed MPhil in Physiology from Mymensingh Medical College. Now I would like to request to her come on dais and request topics, please. Thank you.
Thank you, Madam. Bismillahirrahmanirrahim. Assalamu alaikum and very good morning to all. With permission of Chair, I am going to present my topics. And today's topic is cell-to-cell signaling, intercellular communication. These are the contents of today's topics. Introduction, modes of cell signaling, intercellular communication, receptors, signaling pathways, importance and key takeaways. Now, introduction. Cell-to-cell signaling is fundamental biological process that allows cells to communicate and coordinate their activities in multicellular organisms. Cell signaling is a part of complex system of communication that governs basic cellular activities and coordinates cell action. The ability of cell to perceive and correctly respond to their microenvironment is the basis of development of tissues, tissue repair and immunity as well as environmental homeostasis. This figure showing basic difference between the cell signaling and signal transduction. Here, cell signaling is about communication between different groups of cells and tissues, how one group of cells inform another group of cells what to do. And signal transduction refers to how the presence of an extracellular signal can produce a change in the intracellular state of the cell without the initial signal crossing the membrane. And this figure shows the different components of signaling molecules, where this I discuss later. And this is the basic figure and basic composition of all the important composition in cell membrane. Now, types of cell signaling: autocrine, endocrine, paracrine and juxtacrine cell signaling. In case of autocrine signaling, a cell signals to itself, releasing a ligand that binds to receptor on its own surface. Autocrine signaling plays an important role during development, helping cells take on and reinforce their correct identities. Also, in case of cancer, and it's thought to play a key role in metastasis. Example is immunocells releasing cytokines, interleukin-1, produced by macrophages to stimulate themselves. In long-distance endocrine signaling, signals are produced by a specialized cell and released into the bloodstream, which carries them to the target cells in distant parts of the body. Each endocrine glands release one or more types of hormone, and many of which are master regulators of development and physiology. When cells are near one another, communicate through the release of chemical messengers which communicate over relatively short distance. One unique example of paracrine signaling is synaptic signaling, in which nerve cells transmit signals. And in case of juxtakinase signaling, direct cell-to-cell contact via membrane-bound ligands. Example is Notch signaling in embryonic development. And also some adhesion molecules like integrins also transmit signals that influence cell behavior. This figure showing the different types of signaling. Here in case of autocrine signaling, the same cell, target sites are same cell, and paracrine, the adjacent cells, and in case of endocrine signaling, distant cells is the target cell. Here, synaptic transmission occur. Here, neurotransmitter releases in the synaptic cleft, which bind to the receptor in the postsynaptic membrane. And this figure shows the gap junction, which is composed of connexin in subunit, and different types of signaling molecule transmit from one cell to another cell by this gap junction. Now, key components of signaling pathways. Signaling molecules, ligands, protein, peptide, steroid, gases, and small molecules. And receptors, cell surface receptor, G protein coupled receptors, receptor tyrosine kinase, and intercellular receptor like steroid hormone receptors. Also, J-state pathway, where cytokine receptor activate the STAT transcription factors. Example is immunoresponse regulation. Signal transduction pathways, where cascades of molecular interaction occur, and phosphorylation, second messenger are the example. Last of all, cellular response, where changes in gene expression, metabolism, or cell behavior. Now, what are receptors? Receptors are proteins associated with cell membranes or located within the cell. They recognize signaling molecules by binding to them and causes cell behavior changes. These are the three types of receptor categories: G protein coupled receptors, ion channel receptors, receptors with intrinsic enzymatic activity like receptor tyrosine kinases. This figure shows three types of receptors. Intracellular signal transduction pathways. The chains of molecules that relay signals inside a cell are known as intracellular signal transduction pathways. When a ligand binds to a cell surface receptor, the receptor's intracellular domain changes. Generally, it takes on a new shape which may make it active as an enzyme or let it bind other molecules. This change in the receptors sets off a series of signaling events. For instance, the receptor may turn on another signaling molecule inside the cell, which in turn activates its own target. This chain reaction can eventually lead to a change in the cell's behavior or characteristics. And these three stages of signal transduction, where reception occur and receptor bind with the signaling molecule, transduction pathway, where series of events occur in signal transduction pathway, and last of all, activation of cellular response occur. Ligand-gated channels are ion channels located on the postsynaptic side of the neuron. Some act in response to a secreted ligand and neurotransmitter like acetylcholine, GABA, glycine, glutamate. Some acts in response to internal ligands such as G proteins. Cyclic AMP, cyclic GMP are also regulated by internal metabolites such as phosphoinositides, arachidonic acid, and calcium. Now, intracellular signaling after hormone receptor activation, which form by hormone receptor complex. Here is example for ion channel link receptor. These are the pathways for ion channel receptor that I showed before figure. Here neurotransmitter acetylcholine release combines with the receptor in the postsynaptic membrane, changes in the structure of the receptors by opening or closing of ion channel. G protein link hormone receptor is very important mechanism that indirectly regulates the activity of target proteins. Here, enzymes coupling with groups of cell membrane protein named heterotrimeric guanosine triphosphate. And it has three parts: alpha, beta, and gamma subunits. And ligand bind to extracellular part of the receptor, where a conformational changes occur which activates the G proteins and induces intercellular signals that either open or close cell membrane ion channels and change the activity of enzyme in the cytoplasm of the cell or activate gene transcription. Here is the mechanism where exchange of GDP for GTP, and this causes alpha subunit to dissociate from the trimeric complex, and where alter the activity of ion channel or intercellular enzymes like adenylyl cyclase or phospholipase C. But when hormone removed, the all action reverse and GTP converted into GTP again. These are the key components of this mechanism, and this figure showing all the mechanism that I described before. This is also a figure of G protein coupled receptor. Now, intracellular hormone receptors and activations of gene. Steroid hormones, thyroid hormones, retinoid and vitamin D, they are lipid soluble, cross the cell membrane and interact with the receptor, binds with the DNA strands in the chromosome, activates transcription process to form mRNA. And this mRNA promotes translation, which causes new protein synthesis. This figure shows all the mechanism that I described. Here, hormone receptor complex bind with the DNA strands and produce mRNA, which produce protein synthesis in the ribosome. Now, enzyme-linked hormone receptors, where peptide, growth factor, cytokines, hormones that use this enzyme-linked receptor, like tyrosine kinase, cell signaling. In case of this signaling, tyrosine kinase of the genus kinase family, JAK2, which causes receptor JAK2 complex and causes phosphorylation of STAT proteins, activate transcription, which initiate protein synthesis. These are the hormones that use this tyrosine kinase signaling. And this is a figure showing the JAK-STAT signaling, where leptin receptor act by this mechanism. These are the other second messenger mechanism for intracellular hormonal function. Here, adenylyl cyclase, cyclic AMP second messenger system initiate a powerful cascading activating force for the entire cell. Cell membrane phospholipid second messenger system activate enzyme phospholipase C, which catalyzes the breakdown of phospholipids, phosphoinositol biphosphate, inositol triphosphate, and diacylglycerol. Calcium calmodulin second messenger system also causes activation of calmodulin dependent protein kinase, causes phosphorylation. And specific function of this calmodulin is activate myosin light chain kinase, which acts directly on myosin of smooth muscle to cause smooth muscle contraction. These are the hormones that use adenylyl cyclase, cyclic AMP second messenger system. Now, importance of cell signaling. Coordination and regulation. Here, multicellular organism acts as cohesive unit, development and differentiation, response to environmental changes, disease prevention and treatment, metabolic control, and also immunoresponse. These are some examples of dysregulation in the cell signaling pathway. In case of ion channel, various neurological disorders may occur, and also muscle disorder, stroke, multiple sclerosis, Alzheimer's disease, myasthenia gravis. And when mutation occur in G protein coupled receptor, many kinds of disease like acquired and inherited disease, retinitis pigmentosa, hypo and hyperthyroidism, nephrogenic diabetes insipidus, also even carcinoma may occur. And in case of enzyme link, inflammatory and autoimmune disease, rheumatoid arthritis, psoriasis, inflammatory bowel disease. And second messenger system dysregulation causes neurological and neurodegenerative disorder, including Parkinson's disease. Now, key takeaways. Cell-to-cell signaling is a language of cells, which is complex yet highly regulated process. Understanding these mechanisms helps in developing therapies for disease caused by signaling dysregulation. Cell errors in cellular information processing are responsible for diseases such as cancer, autoimmunity, and diabetes. By understanding cell signaling, diseases may be treated effectively and artificial tissues may be created. And thank you all for patience here.